Hello, my name is Lisa Smith and I'm a Professor of Criminology at the University of Leicester. In this training video, I'm going to show you how to use DNA kits developed by a research team at the University of Leicester in collaboration with the Wangu Kanja Foundation and Kenyatta University with funding provided by the Ring for Peace Foundation. In this presentation, we are going to have a look at how these DNA kits can be integrated into existing practice at healthcare clinics in Kenya. And I'm going to show you the components of the kit and discuss how the design of the kits can improve the collection, preservation, and integrity of DNA evidence to support investigations and prosecutions of sexual violence. So in this presentation, we're going to talk through a summary of the DNA evidence kit project and the approach that we're taking with these kits. We're going to talk through the contents of the kit piece by piece so that you can really clearly understand how to use them and how easily they can be embedded in your existing practice in healthcare clinics in Kenya. And that's going to consist of talking through two components mainly that you would use with patients in a healthcare setting. The mouth swab that's used to collect DNA from the patient, and then the vaginal swab, which is used to collect DNA evidence that might contain the perpetrator's DNA after an incident of sexual violence. So how to use that swab properly and how to package it properly. And then finally, we're going to look at the overall packaging for the kit, which is an evidence bag, which will help you to preserve evidence as best you can and ensure that chain of custody is maintained along the way. So the DNA evidence kit sample collection, this kit that we're going to talk through today is suitable for use with adolescent and adult female survivors who are reporting sexual violence to your clinic, where the perpetrated sexual violence that they're alleging occurred includes vaginal penetration. Now, you can basically use this DNA kit with any patient that you would usually carry out a vaginal swab for. So anybody that is of a suitable age for a vaginal swab, you can use this kit in those cases in place of what you're already doing. Ideally, you want to be using this kit within 72 hours of the incident of sexual violence occurring. And this is because we know that as time goes on after an incident of sexual violence, the likelihood of successfully recovering DNA evidence diminishes a lot over time. So patients ideally need to have their medical examination done as quickly as possible after sexual violence has occurred. And any time after 72 hours, you're very unlikely to get a successful DNA swab from a vaginal swab. So ideally, you want to be using this kit within 72 hours, but ideally much less than that if possible. Now, in our work that we've done in Kenya so far, we have visited a lot of clinic settings, and we know that a lot of clinics are doing vaginal swabs already. And in many cases, they're doing 2 or 3 different swabs. One swab usually for the purpose of collecting forensic evidence that would hopefully be picked up by the police and processed by the government chemist. And then another 1 or 2 swabs, vaginal swabs, which are then sent off to the hospital lab for other microbiology analysis, for example, looking at the presence of STIs. Now, this swab that we're going to talk through today, which is for the purpose of forensic DNA evidence, should be carried out before you take the other vaginal swabs that are sent off for microbiology testing in your clinic or in your hospital. And the reason we're recommending that you use this the kit in this swab before those other microbiology swabs is that you don't want it. There's every chance that there's going to be very little DNA evidence from the perpetrator of the sexual assault, and so you really want to enhance your chances of picking that DNA up on the forensic evidence swab, which is why we are recommending that you do that swab first so that you don't potentially lose out on the opportunity to find that DNA from the perpetrator. So we are recommending that you use the swabs in this kit first and then go on to do the vaginal swabs that you would do for microbiology testing. Now, one of the new and kind of innovative things about this kit is that you have the option to either use this kit as you would normally carry out your vaginal swabs for forensic evidence recovery, or the kit can be used in a supervised self-examination sense. So in other words, the patient could be given the opportunity to use the 2 swabs in this kit themselves under your supervision, so within the clinic and with your guidance and help, but giving them the opportunity to take some control of the vaginal swab and actually carry it out on themselves. And this can really enhance the patient experience. For some women, this may make the exam feel a little less invasive, and it also may make them feel quite empowered to be taking some control over their own bodies and over the search for evidence from their own bodies during the examination. So we would recommend that you make that option available to patients and do whichever version of it that makes them feel the most comfortable. Now, just a quick summary, and hopefully this is something you already know about, but just a quick recap on why we're doing this kind of DNA swab from a criminal justice system point of view. So the DNA evidence kits are used to recover and preserve DNA evidence for collection by police or the courts. And DNA in cases of sexual violence is really most useful in cases where the survivor is not in an intimate relationship with the alleged perpetrator. So where they're reporting that the abuse that they suffered was carried out by somebody that either they don't know, so somebody who's a stranger, or somebody who is perhaps known to them, but they're not in an intimate relationship with. So like a teacher or a pastor at the church or a neighbor, something like that, because DNA will not help the criminal justice system determine whether sexual contact between 2 people was consensual or not. But what it does help determine is whether or not 2 individuals had sexual contact with one another. So it's very useful for cases where you need to identify who the person was that had sexual contact with the survivor. So in other cases, it's also useful where the survivor is a child, because by law, children can't give consent to sexual violence. So regardless of who the perpetrator was, whether it's somebody known to them or not, being able to establish that a particular individual had sexual contact with a child is always useful from a criminal justice point of view. And DNA in a criminal justice setting is really valuable because it can help, as I said before, to identify a particular individual. So to give the police and the courts confidence that their suspect, let's say, is in fact the person who had sexual contact with the victim. Equally important, though, is it can also provide really strong evidence to exclude a particular person. So to say that a suspect perhaps is definitely not the person who had sexual contact with that survivor, which is equally important in cases where perhaps the police have arrested the wrong person. So not only is it really useful for identifying the guilty perpetrator, it's also very, very good at excluding somebody who's actually not guilty of a particular sexual offense. So those are a couple of the key reasons why it's important to collect forensic DNA evidence in cases of sexual violence. And this kit is going to help you to do that in a really best practice, kind of gold standard sort of way. Okay, so the kits that we're talking through today look like this that you can see on the screen. So they come in outer packaging, and inside that outer packaging, you will find 2 swabs, one for the mouth and one for the vagina. You'll also find a pair of gloves and an evidence bag inside. So this is what the kit components look like when you've opened that outer packaging. So you can see the pair of gloves, the evidence bag, and 2 swabs. And to note here, the 2 swabs are color-coded. So one is a yellow capped swab, which you can see on the far right there, and the other is a red cap swab. The yellow swab is the one that you use to take the mouth sample from the patient, and the red swab is the one that is to be used for the vaginal swab. So first we're going to talk through how to carry out the mouth swab properly, which is the yellow cap swab in the kit. So this is what the mouth swab looks like when you take it out of the kit and open it. So on the left-hand picture, that's what it looks like while it's still sealed. And on the right, you can see that the yellow cap end is the end that has the swab attached to it. And then there's also a black cap on the opposite end of the tube that can also be removed. And we're going to see why and when you remove the black cap very shortly. So you'll also find that in your kit there's an instruction manual. And we're going to look at some screenshots of the instructions as they appear in the instruction manual. So just a reminder, the instruction manual is written from the point of view of the survivor. So if the survivor would like to carry out these swabs themselves, then the instruction manual talks through how they should do that. If they prefer the clinician or the medic to do it, to do the examination for them, the same instructions should be followed, but from the medic's point of view. So this is what the steps for the mouth swab look like in the instruction manual that you'd find in the kit. And so the first step here is to open up the outer packaging, as we've already seen, and take out the yellow mouth swab. Now, you remove the mouth swab from its tube very easily by holding the yellow end and twisting it and removing it from the tube that it's packaged in. And then while holding the yellow end, the swab is then placed in the survivor's mouth, and you rub the inside of the cheek for about 5 seconds on each side of the mouth with the swab in order to recover the DNA sample. Now, it's really important to remember that the purpose of this swab is not to collect saliva. When we have done some field work in Kenya as part of this research project, when we visited the government chemist, we were told on numerous occasions that they sometimes get mouth swabs or buccal swabs, as they're called, which do not have a viable DNA sample on them. And one of the reasons they think this might be a problem is that the swabs are being taken in a way that just attempts to collect saliva. But what you actually want to do with this swab is collect cells from the inside of the cheek. So the swab does have to be firmly but gently rubbed against the inner side of the cheek for a number of seconds on both sides to hopefully get a combination of saliva and cells from the inner cheek. So it's really important that if the survivor is doing this themselves, you explain to them that they should be pressing the swab up against the inside of their cheek while they rub it for 5 seconds on each side, or if you're doing it as the clinician for the patient, to make sure that you are in fact in contact with the cheek. It can sometimes be helpful to, if you're administering the swab, to put your other hand on the outside of the cheek, and you should be able to feel the swab moving around inside the patient's cheek from the outside, just to make sure that you are in fact making sufficient contact with the inside of the cheek. Once you've done the swab for a few seconds, for about 5 seconds on each side of the cheek, the next steps in the instruction manual show you then how to repackage the swab. So you place the swab back inside its tube that it was originally in, press the yellow end firmly to click it shut, and then now you can take a pen or a marker and label the outside of the tube. So it's a good idea to label it with the type of swab that was taken, so patient mouth swab, the date that it was taken, any other information that you would wish to record in your normal practice. So it could be the name of the survivor or a case file number, perhaps the name of the clinician. Whatever you would normally record on swabs can be recorded on there now. And that's it for the mouth swab. So you can then put the used tube, now that it's packaged back in its tube, you can put the used swab to one side while you move on to the vaginal swab instructions. So before we move on to talk about the vaginal swab, I just wanted to show you the mouth swab in use. We've just talked through how to use this. So this is an example of the mouth swab with the yellow end and the black end. So just to demonstrate this for you one last time, to remove the swab, you hold the yellow end and twist and remove the swab. To use the swab for the mouth swab, it goes into the survivor's mouth. Either they can do this to themselves or you can do it for them as the clinician. Note that you don't need to be wearing gloves for this step, or if the survivor is doing it themselves, they don't have to put gloves on yet. If you're doing it for them, then you should be wearing gloves as you would normally be wearing as a clinician. But if the survivor is doing it themselves, they do not need to put the gloves on for this stage. So the survivor is going to use the swab to rub it on the inside of their cheek for about 5 seconds on each side. When they're done, the swab goes back into the tube and you push the yellow end in to secure it. And then you can put this swab aside for now to move on to the vaginal swab instructions, which we'll talk through right now. Okay, now that we have looked at how to use the mouth swab, we're going to move on to talk through how to use the vaginal swab. So just going back to the kit components again in this picture, we are now going to move on to look at the red cap swab, which is used for the vaginal swab. So this is what the swab looks like when it's removed from the outer packaging. It looks much like the yellow swab, except it has a red cap. And the other thing you'll notice is that it is a slightly shorter swab than the yellow swab. Now, the reason it's a little bit shorter is because this kit is designed to be able to give you the option to let the survivor do a supervised self-examination. So what we want is to make sure that to decrease the chances of them injuring themselves by inserting a swab too far into their vagina. So this swab is a little bit shorter so that it is safe to insert as far as is comfortable up to the red handle of the swab. And it mimics what the results you would get from a high vaginal swab when it's being done without a speculum. So we have visited a fair number of clinics in Kenya, and a number of them told us that they either never use a speculum in cases of sexual violence, or they often don't use a speculum for various reasons. Some clinics felt that the use of a speculum was too traumatic and too invasive for survivors of sexual violence. Some clinics have told us that most survivors decline the use of a speculum, and so in cases where you're not using a speculum, what you're essentially doing is a blind vaginal swab, and that's exactly what this swab will do. But it also will enable you to either carry out that blind vaginal swab for the patient or to let the patient carry it out themselves. If you are in a situation where you would normally use a speculum and the survivor is consented to the use of a speculum, you should still do that. And you can use this swab in conjunction with a speculum, but it can be used safely without the aid of a speculum in cases where that's not appropriate. So if we go back to the instructions, this is where it's important that if the survivor is doing the exam themselves, they need to put on the gloves first. So the gloves that come in the kit should be put on before the swab is opened. And once the gloves are securely on, the survivor can remove the swab by holding the red end, twisting it slightly, and pulling the swab out of the tube to release the swab for use. Once they've removed the swab holding the red end, or you've done it if you're going to do the examination as the clinician, this is where it might be useful if the survivor is doing the swab themselves to explain to them that it might be more comfortable for them to either lay down during the examination or perhaps stand up with one leg elevated, but basically they have to have comfortable, easy access to be able to insert the tip of the swab into their vaginal opening and insert it as far as it will easily go. It's also important to tell them to part the lips around the entrance of the vagina as best they can, because what you ideally want to do is try to ensure that the swab doesn't come in contact with too much of the external surfaces of the genitals as it's being inserted and removed. And this is because if DNA from the perpetrator is successfully recovered on this swab, you want to be, the criminal justice system wants to be able to say that that was probably found on there because there was vaginal penetration. So it's important that the swab comes in contact primarily or entirely with the inside of the vagina rather than the outside. So parting the lips around the entrance of the vagina is important. So once the swab is inserted as far as it will easily go and comfortably go, It should be moved in a circular motion for about 10 seconds before being removed. If the swab is not sliding in easily, it can be gently rotated, but again, it's important to ensure that if the survivor is doing this themselves, they don't force it to go any further than is comfortable, because we don't want there shouldn't be really any discomfort from the use of this swab. So it's important that they understand that they shouldn't put it in any further than it will comfortably go. Similar to the yellow swab, once it's been removed from the vagina, so now it's used, it needs to be put back in the tube in the same way as the yellow swab. So inserting the swab back into the tube and pressing the red end firmly to close it. Once again, the supervising clinician should then label the outside of the tube. Similar to the mouth swab, it may be useful to write that it was a vaginal swab, the date, and any other information you would normally record, the name of the survivor perhaps, a case number, the name of the clinician, whatever else you would normally want to be recording on the swab tube, you can record on this tube. Okay, so now that you have used both swabs, you've used the mouth swab to get some DNA, a DNA sample from the survivor from the inside of their cheeks, and you've used the vaginal swab to recover DNA evidence that might be present that may contain DNA from the perpetrator. It's time to package it so that it's ready to be collected by the police or the courts. So also inside that outer packaging, you'll find a breathable evidence bag. And what's going to go into this bag is the two swabs, the yellow tip and the red tip swabs. Now you'll notice in this picture, there's an arrow pointing to the other end of the swabs where the black caps were. And that's because we need to, before we put the swabs in the bag, we need to remove the black cap from both swabs. Now, the reason this is really important is that these swabs and the bag that they're packaged in are designed to allow them to air dry after they've been packaged. But it's only going to be possible for the swabs to air dry within the bag if some air can get into the tube. And so that is why you remove the black caps at the end of the swab. So you have to remove the black caps from both ends of the swab and place the open tube, as soon as you've removed the black cap, immediately into the breathable evidence bag. And you can then throw the black cap away in a rubbish bin. You're going to repeat that with the red cap tube, or whichever tube you remove the cap from first. It doesn't really matter, but just make sure that you remove the black cap from both tubes in turn and immediately place each one inside the evidence bag. Now, it's really important that once both tubes are in the evidence bag and the black caps have been removed, that you then seal the bag properly. So the bag comes with an adhesive strip along the top, so a sticky side along the top. And to access that adhesive strip, you have to remove the blue plastic film from the top of the bag. So you'll see that when you're looking at the bag from the outside, there's an orange strip along the top of the bag. And the inside edge of that orange strip is a blue piece of plastic that can be removed entirely from the top of the bag and thrown away. And that then exposes the sticky seal, which can then be firmly closed by pressing across the length of the opening so that the bag is completely sealed across the top. So just to make sure that that was clear in the instruction illustrations in the slide, I'm just going to show you very briefly how to seal the evidence bag. So this is a breathable evidence bag similar to the one that is in the kit. This is the orange strip across the top that I was talking about. And the blue strip that you want to remove is on the inside surface of that orange strip. So let me just grab the end of it here so you can see what this looks like. Hopefully you can see there that there's a blue strip that's coming off of the inside. You will peel that off entirely. It'll come entirely off the bag, and then you can press across the top of the bag to seal it shut. What that essentially does is it seals the top of the bag in a way that makes the bag tamper evident. So you cannot reopen that seal once it's been sealed, once the blue plastic has been peeled off and it's been sealed. And the reason that's important is that when this bag goes into evidence at the police need to be reassured that nothing has changed inside this bag after it's been sealed by the clinician. And by having tamper-evident packaging, it reassures them that actually nobody has subsequently broken into this seal and perhaps taken something out of the bag or swapped something around in the bag. They can be reassured that everything in the bag is in the same state it was in at the time that the seal was sealed, if that makes sense. The last thing I wanted to say is that I mentioned that before the tubes with the swabs in them go into this bag, the black cap needs to be removed from both tubes. If you were to put the tubes into the bag and seal it shut and then realize you forgot to take the black caps off, it's not the end of the world. Even with the bag sealed, you can maneuver the bag in such a way that you can grab the black caps from the tubes that are inside and still pull them off. And you can just leave the black caps floating around in the bag. That's fine. It's more difficult to do it that way because the bag is a bit slippery. But worst case scenario, the important thing is that the black caps are not securely in the tubes anymore when this goes off to the police so that they can breathe. Now, the interesting thing about these evidence bags is once you've taken the black cap off the tube and you've sealed the bag, this side of the bag, the white side, is breathable. So air is able to permeate in and out of the bag, allowing the swabs inside to continue to dry while they're packaged. So they're protected in there while they're drying. There's no need for open air drying of the tubes. They can go straight into the bag, sealed. As long as the black caps are off, they will breathe and dry within this bag indefinitely. Okay, so now you have used the mouth swab and the vaginal swab. You have removed the black caps from both of the tubes. The tubes have been put into the evidence bag and the evidence bag has been sealed. So that evidence is now secure and safe and able to store at room temperature or refrigerated if you wish, indefinitely until it's collected by the police or the labs or the courts. The last thing you may wish to do is to record some information on the front label of the bag. What I would encourage you to record on there is anything that you would normally record on evidence bags in your practice that you already do. So that could be things like the date and the time that the swabs were taken, the name of the patient or the case file number of the survivor's case, perhaps the location, so what hospital, the name of the clinician or practitioner who supervised the evidence recovery, and anything else that you want to add. There's plenty of room on the front of the bag for you to write whatever information is necessary. Okay, before we wrap up this training session, I just wanted to talk through how these DNA kits improve on some of the current practice that we've witnessed in Kenya during our fieldwork. We visited a number of clinics during some fieldwork in February of this year, and we saw some really great gender violence clinics within hospitals who are doing great work supporting patients and survivors who come in to report sexual violence and get a medical examination. But we did identify some gaps in current practice which could and really should be improved upon in order to increase the integrity of DNA evidence that's being collected. So I'm going to talk through each of these three improvements in turn and hopefully show you how you can integrate these new kits into your existing practice, but make some really positive improvements to how you're collecting DNA in your clinics now. So the first is, as I mentioned, these kits give you the option to offer to the survivor for them to do a self-administered examination under the supervision of a clinician. So this provides survivors with the option to make the procedure more comfortable and less intrusive and somewhat empowering to them if they feel that those things would be achieved by them doing the examination on themselves. If for some people that might not be the case, for some people they would feel more comfortable if the clinician does it, and that's perfectly fine. But for people who might want to be more actively involved in the recovery of evidence, it gives you that option to let them do it under your supervision. The other important thing about this approach and the option to do self-administered examinations is that it doesn't necessarily require a specially trained forensic medical examiner to administer the kits. And the reason why this is important is because our longer term aim with these kits is to be able to make them available in really very low resource clinics. So you could imagine things like in humanitarian emergencies, in displaced communities, in refugee camps, where there isn't a forensic medically trained examiner on site. The idea is that the instructions have been developed in a way that is clear enough that somebody with no forensic training could easily supervise somebody doing a self-examination using this kit. So in your clinic, it might provide survivors with the option to do one, but in other even lower resource settings, it might be the only option. And so that's why we think the self-examination approach is particularly useful. The second improvement that these kits can provide to current practice is the evidence bag. So we've just seen the breathable evidence bag and how it enables the swabs to dry after they've been secured within the bag. And this is really important because we have seen a number of clinics have showed us how they dry their vaginal swabs on site. And the first thing to say is that it's really good practice that these swabs are being dried already in current practice. But what was worrying to us about the current practice is that the way that clinics are drying swabs at the moment really opens up risks of contamination and tampering and human error, which we cannot have in a criminal justice forensic evidence setting. So this is a picture from one of the clinics we visited. All the clinics we visited were doing something similar to this, where they were air drying the swabs in a drying cabinet for anywhere from 2 to 4 days before they were being packaged. So a couple of things to note here. We have a whole bunch of swabs from different cases air drying in the same cabinet next to each other. And this is fine in terms of it will in fact air dry the swabs, but from a criminal justice and forensic point of view, it doesn't protect against contamination. So those swabs are very close to each other in an open air cabinet and could easily come into contact with one another and cross-contaminate. It could also be contaminated by DNA in the environment within that clinic and be picking up DNA from people who had nothing to do with the swab. There is, of course, if somebody wanted to tamper with these swabs, it'd be very easy to do. The cabinets that we saw were not locked in any way. Somebody could easily walk in and either take a swab, so it would then have gone missing, or replace a swab with a different swab. And so tampering could be very, very easily done. And then human error is also something that we have to try to minimize in the forensic sciences. Human error happens in every setting, no matter what. But we should try to reduce the likelihood of human error in a forensic environment. And the problem with air drying the swabs altogether like this is that it's quite easy for somebody to accidentally package one of those swabs in a tube that belonged to a different swab, if that makes sense. So now you've suddenly got a swab packaged with the wrong information written on the outside of it, and suddenly it's not the right swab for the right case. So this is really not a secure way to air dry swabs. And so the evidence bag allows the swabs to air dry after they've been securely packaged. So none of those things, contamination, tampering, or human error, can happen once that bag is secured. And yet the swab can still dry in there indefinitely. So this is a really important development that we think will really improve the way that DNA evidence is dried in your clinic. The final thing to just note in this picture is that most of the swabs that we saw in use in clinics across Kenya were swabs that were on a wooden shaft, like the ones in these pictures. There is international guidelines for forensic swabs state that we should not be using swabs with a wooden shaft for inserting into any human orifice. They're not safe to use as for internal examinations, partly because they can easily break and also because they are wood, so they can splinter. So we really best practice says that we should be using swabs with a plastic shaft, which is why the swabs in our kits here have got a different material for the shaft. They're not wooden. The other thing that we noticed when we were at clinics is that clinics were using sterile swabs, so medical grade sterile swabs, which is great. But from a forensic point of view, for the swab that's used for forensic evidence, we really should be using DNA-free swabs. So swabs that are not just sterile, but are guaranteed to not have DNA on them before they've been opened. Because again, we don't want DNA that has contaminated a swab in a swab where it's going off for forensic testing. So the swabs that are in the DNA kits that I've showed you today, are all certified DNA free. So they're manufactured in a facility that guarantees that there is no human DNA on the swab before it's opened in your clinic. So just two other things, the wooden shaft and the DNA free, are additional improvements to current practice. And the last improvement to current practice is the fact that not only does the evidence bag allow the swabs to air dry and breathe inside of them, but the seal on the bag is also tamper evident. So this is another picture from a clinic that we visited in February, where they had evidence bags in a refrigeration unit, and you can see that, probably similar to whatever clinic you work at, the evidence is being packaged in paper bags or paper envelopes. A lot of the clinics we saw were sealing these paper bags and envelopes with tape and then signing on the tape as a kind of tamper evident method to ensure that if somebody broke the seal of the tape, it would then break the signature and it would be obvious that somebody had opened the bag. That's a good start, but it's not difficult for somebody to tamper with a paper bag, even with the tape and the signature on it. And we know that in forensic evidence, the chain of custody is vitally important. It's very important that the integrity of evidence that's in an evidence bag is maintained from the time it's collected all the way through until it gets to a lab and is tested. So the current use of paper bags and paper envelopes really offers no robust protection against tampering. And the evidence bag in these DNA kits that I've showed you today cannot be opened once they've been sealed without evidence of tampering. So once the bag's been sealed, if somebody tries to open that top of the bag, the orange seal, the words void will appear across the top. So if a lab was to get an evidence bag that had been reopened at some point, it would be very obvious that it's been reopened, and they would be able to note in their files that they didn't process it because it had clearly been tampered with. And so this is a really big improvement on giving confidence to the police and the courts that evidence is being securely packaged while it's being transported from the hospital to the police and to the labs for processing. And as I said in the training, there's also plenty of space on the front of the bag to record all the required information. So in addition to the information you might fill out as a clinician, it can also be filled out with the details of the police officer who collected it and the date it was collected. It can then record any time that evidence bag has changed hands. That transfer can be recorded on the bag. So the courts then have a really clear chain of custody of that piece of evidence, and they can spot any periods of time where the evidence is not accounted for. And that's a really, really important thing to be able to show about forensic evidence. That brings an end to this training video. I hope you've enjoyed hearing about how the DNA kits work and how they can be embedded in your current practice within healthcare clinics in Kenya. If you have any questions or comments for the project team, please feel free to contact us using the email addresses on the screen. So my email address is lisa.smith@le.ac.uk, or you can also contact Wangu Kanja at wangukanja@gmail.com. So we look forward to hearing any feedback that you have or answering any questions you might have. And thanks very much for taking the time to watch this video.